Hey guys, welcome to a lecture on prokaryotic control of gene expression. This is our lecture 2 on genetics of bacteria. So hopefully this picture might look somewhat familiar, and this is the trip operon. And we're going to go into how these processes are regulated. How are genes turned on and turned off in bacteria, basically. We will also be talking about eukaryotic genes, but that will be another lecture. All right, so once again, what does the control of gene expression do? It enables individual bacteria to adjust to environmental change. They can basically turn on a gene and create a protein that helps them to uh, meet the needs or the demands of that environmental change, or they can also adjust enzyme activity. And if you remember from cell signaling, these are also the two major things that happen with cell signaling. We can either turn on a gene, create a protein, or we can adjust the metabolic activity within the cytoplasm of a cell by adjusting enzyme activity too. Okay? So this adjustment of enzyme activity is generally by negative feedback inhibition. Okay, and the tryptophan biosynthesis pathway demonstrates both levels of these controls. Okay, and this should look familiar, this diagram. All right, so remember once again, the trip operon has 5 genes in it. Each one of those genes encodes an enzyme that plays the role in converting a precursor molecule into the amino acid tryptophan. Okay, but if there is plenty of tryptophan available in the environment, why go ahead and make the enzymes that are going to synthesize tryptophan? That would be a waste of energy. So what the bacterium, what has evolved through the process of natural selection, is this negative feedback control where tryptophan, the product of this reaction, if it is abundant in the environment due to diet or something of that nature, then that tryptophan molecule will dock into an allosteric site in the enzyme 1 and shutting the active site down, which then prevents the further uh, um, transcription and translation of the other enzymes and stops this whole process. Again, to save energy. That's how these pathways have evolved. Okay. Um, uh, this Operon system was first described in 1961 by two French biologists who also had a very interesting history as French resistance fighters during World War II. They're pretty wild guys, by the way. And when they described the operon, they came up with 3 elements. The genes that operon controls in prokaryotes. Generally, these genes are grouped together that play a role in either breaking something down like the lac operon or synthesizing like tryptophan in the trip operon. Those genes are together and transcribed and translated as a unit. So we have the genes, we have the promoter region. Again, this is where RNA polymerase is going to bind to begin that transcription process, and the operator region, which is the on-off switch of an operon. And this is where the regulatory protein will bind to in order to shut that operon down, or it will fall off in order to turn that operon on. So let's look at the trip operon right here. And the trip operon is known as a repressible operon. Okay, in this particular picture right here, the trip operon is on. As you can see, the regulatory protein is which is called the inactive repressor, is not bound to the operator, so RNA polymerase is free to bind to the promoter region, transcribe all of those 5 genes, which will then be translated into the 5 different enzymes of this biosynthesis pathway. So the tryptophan is absent, the repressor is inactive, and the operon is on. Now, if tryptophan is present in the environment, do notice here's my tryptophan, also called a co-repressor. It will bind to the inactive form of the repressor protein or the regulatory protein. That's going to cause the repressor protein to go into its active shape, and then it will now bind to the operator and it will shut that down. So you may want to click on that little trip operon animation. It's a little bit more complex than I'm showing here, but that is okay. Again, it is a repressible operon. The trip operon is a repressible operon. This was model 2 in your POGL that you did the other day. And again, it is inhibited when a small molecule binds allosterically to a regulatory protein. That small molecule would be tryptophan, okay? Um, Whereas an inducible operon, one that is only turned on, is stimulated when a specific small molecule interacts with a regulatory protein, okay? So let's go ahead and we will look at this particular example in the lac operon. This was model 1 in the one that you did yesterday. All right, so notice that the regulatory gene codes for an active repressor in this case. In the repressible operon, the regulatory gene coded for an inactive repressor. So the active repressor right here will bind to the operator, and this system is shut down. Okay, and look at our environmental conditions. Lactose is absent, the repressor active operon is off. And what does the lac operon do? It plays a role in producing 3 enzymes that are going to help break down and transport lactose into the cell. Okay, so if there's no lactose present, why bother? It's a waste of energy. So the the regulatory protein is produced in its active repressor form. Now, when lactose is in the system, okay, when lactose is in the system, here we see lactose present. Okay, lactose will bind allosterically. It's called allolactose here, which is just an isomer of lactose, will bind allosterically to the regulatory protein, turning it into an inactive repressor. It has changed shape. This changed shape can no longer bind to the operator. So it will not be bound to the operator. So RNA polymerase is free to bind to the promoter and now transcribe and then translate those genes. Okay, the gene beta galactosidase will play a role in breaking lactose into glucose and galactose. Permease is a transport protein that will allow the faster transport of lactose into the cell. Okay, so that is this operon has been induced by the presence of lactose in order to transcribe and translate the genes that will help to break lactose into a source of energy for the cell. Okay. All right, so you should be able to compare and contrast repressible and inducible operons. As I said earlier, repressible operons are generally on and they're playing a biosynthetic role. They are creating, they're making things, and usually the things that they are making are necessary for the function of the cell. Inducible operons are usually off, and they play a role in catabolic pathways, and again, usually digesting nutrients to simpler molecules. Both of these pathways, however, demonstrate what we call negative control because the active form of the repressor will inhibit transcription. And the active form of the repressor inhibits transcription. So here we see the active form of the repression in the lac operon. It is inhibiting transcription. Here we see the active form of the trip operon. It is inhibiting transcription. So that's why we call that negative control. Now, there is also positive control, and positive control also plays a role in the lac operon. So it's a little bit more complicated than we showed you earlier. And we call it positive control because the active form of a protein, in this place it's called CAP, okay, induces RNA polymerase to bind to the promoter region. And without that active protein, then CAP will not bind. And even if the lac repressor is in its inactive form, i.e., it is not bound to the operator, we will not transcribe and translate those particular genes. Okay? So in this case, what we see here is that there's lactose is present. There's very little glucose here, and therefore these genes, RNA, the cap protein is active. It binds to the promoter region. That's causing RNA polymerase to bind and transcribe those genes so it can utilize lactose as an energy source. But as I say up here, glucose is the preferred food source. Why? Well, glucose is already a monosaccharide. Lactose is a disaccharide. So the cell has to expend more energy to break down lactose and convert it to ATP than it does glucose. So if both are present, it wants to use the glucose and not the lactose. Okay, and high levels of glucose are going to produce a cyclic AMP, which binds to this inactive cap, activates the cap. Now the cap protein binds to a region near the promoter, at the front of the promoter, which will induce RNA polymerase to bind, and boom, there we go. We make all the genes necessary because there's no glucose in the system. I should say low levels of glucose cause this to happen. Okay, low levels of glucose cause this to happen. All right. However, when glucose levels increase within the cell, notice that cap is now inactive. It is not bound to this particular cap binding site. And even though there is an inactive lac repressor, RNA polymerase will not bind to that particular site. And therefore, we're not going to make those genes that are going to break down lactose. Why? Because there's lots of glucose. The cell wants to use the glucose. That helps it to save energy. And that's it. That's the end of that particular lecture, and hope you were able to get something out of it.